The second learning objective is explain how various viruses evade host immune responses, so escape immune responses. Then responses to viral antigens can cause tissue damage. So during a viral infection there will be a lot of tissue damage. Why is that, and what is the response to that? And correlate the immune response with tissue damage and latent viral infections. Now, normally, whenever there is a viral infection, normally viral infections are quite common, and if you think about this region, Makkah especially, there are thousands of pilgrims coming in, and viral infections are quite commonly seen. And what is very specific or special about viral infection is that if somebody has a viral infection, whenever they, for example, influenza infection, and they cough or sneeze, and if you are at a distance of around 6 feet, you are prone to pick the infection through droplet, nuclei or influenza viruses in the droplet, or the shared things, or the common articles that you use. For example, commonly mobiles, they all can transmit the infection. Now, what do you think your immune system should do in such an instance? When you are exposed to a viral infection initially, what you think your immune system will do? How? Phagocytose the pathogen. Recognize the pathogen. Okay, he is talking about the adaptive immune system kicks in and they kill the pathogen. What else?
Innate immune system will work.
So first thing first, you should start with the innate immune system. So the barriers first will try to protect. Yes, innate cells will direct the job of the adaptive cells. So these respiratory, for example, this influenza virus, where it's going to enter first? Nose, respiratory tract. And first cells that are going to encounter or act as a barrier, which will be those? Mucosa. The epithelium of the mucosa is going to act as the primary barrier or the first barrier. So yes, the barriers come in first, then if somehow it traverse the epithelium, then what's going to come? Yes, natural killer is a good answer because it normally kills the viral infection and it's part of the innate immune system. It's good. All your answers will help you, identify that where is the problem, how you are thinking slightly differently. So bacterial infections are different, viral infections are different. You have all the components of innate adaptive immune system there, but which comes first, how they react to the viral infection, that is slightly different. So it's good. All these answers are good. Natural killer cell will be there. Adaptive cell, cytotoxic cells will be there. Which will do what? We'll see that as the lecture unfolds. What else is remaining? We have not named some other things that should be involved there. Macrophages. Complement system, very good. Neutrophils. So just remember all these names now. We have talked about the epithelial barriers. We have talked about the macrophages, neutrophils, cytotoxic cells, natural killer cells, complement proteins. What else? Dendritic cell. The recognition part is done by the dendritic cell. So I'll tell you today something different about the dendritic cells. So every time immunology is building up on the previous knowledge. So whatever you learned previously will go further a step ahead. So yes, the answer to this first question, what do you think your immune system should do? The innate and adaptive immune system, that's going to work together. So they'll cooperate first to protect. The first thing is to protect, then to contain. So special thing about viral infection is that they are very rapidly proliferating. They multiply very fast. So what the immune system will try? First, as a barrier, don't allow it to enter your body. That's number one. Number two is try to contain it. Don't allow it to multiply, proliferate inside the cells. Then, of course, it has entered the body. The job is to eliminate it from the system as well. So you have to think about these three things. First is to stop, prevent its entry. Number two is to contain it, not allow it to multiply or proliferate. Then finally, whatever is inside the body, you have to eliminate it, clean it up. So this all will be done by different components of the immune system. We'll see how they do it. So the first question, I suppose all of you understood this. Now use your imagination and let's go inside your body and see how does your immune system fight against viruses. What happens first? As I said, epithelial cells in the mucosa, they are the first one of the barriers that will encounter the virus. Now initially, the epithelial cells will recognize it, and what is special about it, that so far we know that the first receptors on the immune cells, which are those? Innate cells have got receptors to recognize the pathogen. PRR, pattern recognition receptors. Now today, what you have to learn further is that PRR are not only on immune cells, they are also present on normal healthy cells. For example, here epithelial cells. So PRR will be on healthy cells as well, and they are also on the immune cells. What is special about viral infections? Anything special which differentiates it from bacteria?
It cannot be treated by antibiotics.
Very good. They cannot be treated by antibiotics. Excellent. These are intracellular pathogens. So they cannot cause pathogenicity or disease outside the cell. They have to enter your body first, then they have to enter the cell, and then they have to take control of the nucleus. So once they do that, then they start multiplying, proliferating, and causing damage or disease. So that is, yes?
They totally dependent on host cell.
Host cell, exactly. That's why they take control of the nucleus.
They are way smaller than bacteria.
Excellent. They are very smaller than the bacteria. So these are the special features of the viruses. So they are going to enter, they are encountering the epithelial cells. So they will first try to invade the epithelial cells. That's why it's written obligatory intracellular microorganisms. Now initially they have started to attack the epithelial cells. Now first the epithelial cell will try to react to that or respond to that, but if it continues, as I said, virus goes inside, take control of the nucleus, start multiplying, and gradually it will overcome the cell. So the epithelial cells, you can see in the first figure, the virus is just one. In the second one, there are a lot of small circles that denotes that the virus has started multiplying. And now the alarm kicks in. The epithelial cells have to press the alarm. And what is that alarm? What is that receptor that we very few minutes ago talked about? PRR, pattern recognition receptors, will be activated, and as a result, what will happen? As a result, Now, how do you think the virally infected cells alert your immune system to trigger the immune response against invading viruses? So the PRR are activated. Epithelial cells have to do something to tell the immune system that there is a virus. Send cytokinase or cytokines. Okay, cytokines, pro-inflammatory cytokines. Can you name any? Excellent. Interleukin 1, interleukin 6, TNF alpha. These are the pro-inflammatory cytokines, and somebody named interferon. So production of inflammatory cytokines and type 1 interferon. These are the cytokines that will be produced. And as a result, what will happen? Induction of type 1 interferon in response to viral infection by which cells? By epithelial cells. Now here I would like you to focus on two important aspects. Number one, the pattern recognition receptors. Where are they present? On innate cells, on their surface, and cytoplasm. Yes, on the nuclear membrane as well. Why are they present there? For viruses. Excellent. If there was no pattern recognition receptor in the cytoplasm or in the, on the nuclear membrane, then how they are going to recognize virus? This is the special quality of virus, that it has to penetrate the cell, go into the nucleus. So in the cytoplasm and around the nucleus, they should be recognized. Now this cell, infected cell, in this particular patient, it is epithelial cell, and it has got the pattern recognition receptor, which is called as RIG1, retinoic acid inducing growth factor 1. So RIG1 will recognize it, and whenever the pattern recognition receptor recognize, they do transcription factor production. So signaling, just for simple reason, they will do some signaling. As a result, interferon alpha and interferon beta will be produced. These are type 1 interferons. Okay, so this is the infected cell. On the other hand, now we see here a dendritic cell. What's the name of this dendritic cell? It's written. Have you heard this name before? No. So dendritic cell, so far we know the classical conventional dendritic cell, its job is to and T cell in the lymph nodes or in the spleen. This is called as antigen presenting cell. Okay? And how do they do it? With the help of which receptors? No, PRR they recognize. How they present it? MHC class 1 and MHC class 2 in particular. Now, plasmacytoid dendritic cells can be produced by the bone marrow. They are called as myeloid dendritic cells. They can be produced by other cells. For example, in the secondary lymphoid organs, they are called as lymphoid dendritic cells. Now, plasmacytoid dendritic cell is a lymphoid dendritic cell. It is not classical dendritic cell. It does not do just the job of antigen presentation, the classical antigen presentation. In fact, it do something different. It will recognize the pathogen with the help of PRR, and in this again, where is the PRR? On the cell surface or inside? Inside. So it has got TLR 7 and 9. It will recognize the pathogen and produce interferon, type 1 interferon, alpha and beta. So you have got now type 1 interferon coming from the epithelium cells, and they are coming from the plasmacytoid dendritic cell. And they will create something special, and that is called as antiviral state. Okay, these interferon alpha and interferon beta, they are going to create antiviral state. So first thing, these are the PRRs, RIG1 in epithelial cells inside the cell, TLR 7 and 9 again inside the cell. They will recognize the virus, nucleic acid of the virus, and these are the pattern recognition receptors, and they will produce the type 1 interferon alpha and beta. What these type 1 interferon alpha and beta is going to do, you see, the source is plasmacytoid dendritic cell, the infected cell, and the job they do is they create antiviral state by doing what? By activating macrophages, natural killer cells, CD4 cells, and conventional or classical dendritic cells, CD8 cells, B cells. So basically they are now telling all the other cells of the adaptive system and of the innate system, macrophages and dendritic cell of the innate system, B cells and T cells of the adaptive system, that the virus is inside your body. So you can now link it. Epithelium is the first barrier. It is encountering the virus. Virus goes inside. PRR recognize it. They ring the bell, start producing interferon alpha and beta by plasmacytoid dendritic cell as well as by the epithelial cells. Then they go and alarm all the other immune cells. Now one thing you remember here is that in viral infection, interferon type 1 is very important, very crucial. We'll see its function, but 50% of that quantity that is produced is by plasmacytoid dendritic cells. Even though the epithelium first encounter these viruses, but plasmacytoid dendritic cell is the major producer or the source of type 1 interferon. So are we all okay with this thing? So who is producing type 1 interferon? Major. 50% of the type 1 will be produced during viral infection by plasmacytoid dendritic cells. Now, we have talked about how much it is produced and which cells are producing it. What it can do, it can create an antiviral state. Okay, so what is that antiviral state? First thing, protect the neighboring cell. So this is the epithelial cell which is infected. It is going to release it around all the neighboring cells which are there, because virus proliferate in one cell, breaks, destroys that cell, and then go and infect the neighboring cell, and that's how it spreads. So I told you that in viral infection, what you have to remember, first thing is to protect its entry by the barriers. Number two, to contain it, because it's multiplying very rapidly, so you have to contain it. So when it comes out of the cell, if it enters another cell, it's going to go there, multiply even more faster and bigger. Then it continues that cycle to the next cells. So first thing, the receptors. So all the viruses, they will enter any cell by the help of receptors. So this is the virus that is trying to enter any normal healthy cell. It will first bind to any receptor on the normal cells. Then it goes inside and then enter what? Nucleus. It takes control of the nucleus. I will tell you certain steps that how, which receptors will recognize it of the immune cells, and which, steps the virus is going to go through, because it will be helpful in the later stage of the, lecture. So virus go into the nucleus. What it does, first thing, it will do transcription. So it will produce a mRNA or, so here is, first step is transcription. As a result, what will happen? Next step, translation. And a protein will be produced. Now this is viral protein. So the virus will enter through a receptor, goes to the nucleus, do transcription, translation, protein is produced. This protein will somehow enter the endoplasmic reticulum. Now this part I am going to link with how this viral protein is presented to MHC class 1. Okay? Where is MHC class 1 present? Which cells express it? No. Which cells produce MHC class 1? All nucleated cells. And today this point is very relevant. Why? Because all viruses, they need nucleus to survive. That's why for their protection, all nucleated cells have got MHC class 1 expression. So here is your MHC class 1 in the endoplasmic reticulum. Then there is a structure in the cells which is called as proteasome. What is its job? It's going to take this viral protein and break it down into small pieces. So it enters from here, and then viral proteins are broken down into small pieces. This viral protein is then entering the endoplasmic reticulum, ER, and then it is loaded onto this MHC class 1, which is already present here. And then finally MHC class 1 comes on the surface with the viral protein, and who is it going to present to? CD8, CTL, cytotoxic T lymphocyte. So just remember this thing, that viruses for entry, what they need? A receptor. Then when they enter, they go into the nucleus, do transcription, translation. The protein is taken up by proteasome, broken down, loaded up onto the MHC class 1, and then this MHC express it on the surface. Cytotoxic T lymphocyte is going to recognize it. But this is the whole thing. At the same time, when virus enter, I told you here in the cytoplasm will be PRRs. Okay, these PRR in the form of RIG, and they will be triggered. They will produce certain transcription. As a result, what is produced? Interferon type 1. Okay, this interferon type 1 is going to come out and act on the neighboring cell and create antiviral state, protect this cell. How? First, by blocking this receptor. So you can see this receptor by which this virus entered, the interferon type 1, it's going to block it here. That's the first thing it does. So if you want to protect the neighboring cell, protect this receptor with which the virus has entered. Okay, so that's the first point here. Number 2, so the neighboring cell bind interferon to their surface, which makes them less susceptible to virus. Yes, please.
CD8 recognize the virus. It will do the same mechanism like perforin and glycine.
Yes, it will. But you know, I put it all together at this moment, but I'll come to it later when the role of CD8 cell come. Now I just want you to see that a normal cell who is infected by virus, it will be first in the cytoplasm, activate the PRRs, produce type 1 interferon. That type 1 interferon alpha and beta comes out, block other receptors trying to protect the neighboring cell. At the same time, the virus is doing transcription, translation, protein function, and it can be expressed to the other CD8 cells with the help of MHC class 1. So there are two ways. One is main presentation. So if you think about logically why the nucleus containing cells have got MHC class 1, because they can be infected by viruses. Then if they directly present to CD8 cell, this CD8 will directly kill virally infected cell, and that is what you want. When we want to eliminate it, we have to kill the cell that has got the virus inside it. But at the same time, some part of it will be broken down, taken up by macrophages, taken up by dendritic cell, and presented to CD4, because you need now antibodies, you need activation of B cells. So a lot of things are happening. Just at the moment, this bit you remember. Now the next stage, what they will do, they will inhibit viral replication in the neighboring cells. How? So there is you saw the nucleic acid messenger RNA transcription, translation that is occurring. This type 1 interferon in this neighboring cell can protect that. So here is the nucleus. Here, when, for example, if virus entered, still they will try to block this transcription and translation process, this with the help of this interferon. So they are now trying to create antiviral state. They are trying to protect it. Now what happens if the viral infection proceeds? So this was not enough. Type 1 interferon was not enough to contain it or stop the replication. They are multiplying more, they are creating now damage. So in the cells they go, they proliferate, they make more copies, destroy that cell, go to the neighboring cell, infect that cell. So it's getting more and more messy. More number of viruses are produced. So what you have to do, now there is a question for you. Name the first immune cell that arrived the site of infection to clean up this mess. No, you can now discuss it with the tutors, because there is something important here. We'll start back. Just to clarify a couple of things, because this is what I found out while discussing with the students. Interferon are of two types. Type 1 interferon is basically alpha and beta. Okay. Type 2 interferon is interferon gamma. That's number one. Number two, this interferon, when I am talking about the antiviral state production by dendritic cells and by the plasmacytoid, I am talking about only type 1 at the moment. This is the initial 2 to 3 hours of exposure to the viral infection. Okay. Secondly, the cell that will arrive, some of the students have said neutrophils, rightly said, because when they think of the bacterial infection, the first cell to arrive is neutrophil. But here the question is different. It is who is going to clean up the mess. So the virus entered the cell, triggered the PRR, interferon production, and neighboring cells were trying to protect it. At the same time, it started to multiply, increase their copies, and when there is lot of virus in the tissue, macrophages are already there to start engulfing or taking up the mess. But the neutrophils have to be called from the blood to the tissue. So the first cell to clean them up, to phagocytose them, is the job of the macrophages. So it's the macrophage that is going to clean up the mess. And what it's going to do, we'll see. The macrophages is going to attract by the type 1 interferon that we have already seen. So the infected cell will produce interferon, and that will attract the neutrophil, and it will start eating. And what it is going to do, first thing, it is going to phagocytose the virus. Then, and also the cells that are infected. So it can phagocytose both of them, freely circulating viruses or inside the cells. Killing of the virus, so it not just takes it up, but it has to kill it. How that killing machinery is activated, it's very similar to the thing that we saw in the bacterial infection, that it takes it up and waits for the order from which cell? CD4 cells will order it with which cytokine? Interferon gamma, type 2. So that adaptive immune system rely on interferon gamma more, especially the CD4 cells. And the production of, now it will start to produce its own cytokines. It will produce TNF alpha, nitric oxide, interferon alpha and beta also it will start to produce. So the macrophages in turn then start to produce a lot of things. But what if it is not able to finish the job? Now he is going to call up more backup, and that backup is in the form of neutrophils. So the macrophage is now going to call the neutrophils. So what happened so far? Epithelial cell is the barrier. It produced type 1 interferon, and then it triggered the activation of the, not activation, chemotaxis of the macrophages, so that they can take up more viruses and the virus infected cells. When the macrophages were activated, they produced TNF alpha, the type 1 interferon, and they further called the neutrophils. Another feature of the neutrophil is that neutrophil come to the site of infection, try to fight, and then after that he dies. Macrophage's role is not finished here. They take up the pathogen, try to kill it until it finishes it. Once the infection is over, then it will help in cleaning the tissue and also in repair and regeneration. So the macrophage role is all the way till the end. Neutrophils, they come acutely, they will fight, and then they will die. Now the next cell, the NK cell, natural killer cell mediated killing of the infected cells. Yes, please.
So aren't neutrophils specific for bacteria?
Yes, they are very specific for bacterial infection, but it does not mean that they do not participate at all in the viral infection. They will do as much they can. Definitely it is quite less in percentage. They are very strongly involved in the bacterial infections. So now the natural killer cells are coming. By what? They will be called by type 1 interferon produced by epithelial cells, plasmacytoid dendritic cells, macrophages, and TNF alpha and IL-12. So they come with their properties. What is the killing property of natural killer cell? No, killing property. How they are going to kill? Granzyme, perforins, what else? Fast ligand, very good. So they are going to do that, and that's why he has a cytotoxin gun. You know, they will kill with its perforins and granzymes. Now 2 days after the viral infection, now slightly we have gone further. So during 2 days, it was epithelial cells, macrophages, neutrophils, natural killer cells. They were trying to contain the situation, and now the cells are more and more destroyed by the viruses. So what's going to happen? In addition to the cytokine, they will produce interferon gamma, and they will try to mediate antibody-dependent cell-mediated cytotoxicity. So first thing is, how can natural killer cell recognize the infected cells? Now you can discuss it with your respective tutors. So there were different answers, but I'll just give you the final answer. That very important thing which is related here, because we are taking the whole story like this, that the all nucleated cells can be infected by viruses, all nucleated cells can express MHC class 1, and that MHC class 1 can be recognized by CD8 cells. So loss a counter system like HIV virus, it switches off MHC class 1 expression. So what will happen then? Then it's the natural killer cell that is the alternate mechanism. So first thing is, absence of MHC class 1 will activate the natural killer cell. That's one. Number two is stress receptors. There is a balance between inhibitory and stress receptors. So when viral infection occurs, stress receptors are expressed. And the third thing is ADCC, antibody-dependent cell-mediated cytotoxicity. Antibody binds to the viruses, activate the natural killer cells, and then they do the remaining job. Now, the killer cells have tried to do their job in the presence of the macrophages and type 1 interferon. As I said, we will talk about the entry, protection, containing the virus, stopping it from multiplying, and what if it goes further? It has entered, it has started to multiply, it has started to infect other cells. So we have to think of further going and activating the adaptive immune system, which can eliminate it. So our job is not done by stopping it, by protecting it, but also you have to finish it out of the body, eliminate the virus as well. So virus elimination is accomplished when virus-specific T cells are produced. So adaptive immune system is very important. Now the question is, if virus-infected cells are present in a patient, which of the following cell population are transformed into cytotoxic T lymphocytes whose function is to directly kill the virus-infected cells? Antigen presentation by the classic dendritic cell will cause activation of CD4 cells, CD8 cells, and B cells, and all of them have got different roles to play. We have already talked about CD8 cells are now, when activated, they become cytotoxic T lymphocyte. They will kill the cells that have got virus in them. B cells, now B cells with the help of CD4 cells are going to produce the antibodies that are specific to the virus, and that will require class switching, somatic hypermutation in the lymph nodes, and CD4 cell will start producing IL-2 and interferon gamma. So CD4 and CD8 T cell recognize the antigen displayed by what protein? So you have got now CD4 population, you have got CD8 population. Both of them will recognize the viral protein with the help of which receptors? CD4 with the help of MHC class 2 and CD8 with the help of MHC class 1. Now previously we mentioned that T CD8 cells play a major role in eliminating the viral infection. What is the role of helper T CD4 in immune response against viruses? So it's very clear that now the specific CD8 cells, they will recognize the MHC class 1, and then they will start destroying those cells by perforins, granzymes, fast ligand. So all those mechanisms will come into play and they will destroy. Now CD4 cells are also activated during this process. What is their job? So you write down and then we will talk. To this question, previously we mentioned that T CD8 cells play a major role in eliminating the virus infection. What is the role of helper CD4 cell in immune response against viruses? So what they will do, they will increase the army of CD8 cells which have been, which have recognized the virus. They will produce IL-2 to increase their numbers. That's one. Then they can also help the B cells in class switching. So they help them class switch for viral infection. Which immunoglobulin you need?
IgA.
IgA mainly, and they will be produced at the secretions, tears, salivary secretions, milk, in the GI tract, all the mucosal surfaces. And what they will do, they will neutralize the virus. So this is one property, that virus need receptor to bind and enter the cell. They will block the receptor binding portion of the virus, and this is called as neutralization. They will neutralize the virus so that the virus cannot enter the cell. Okay, so this receptor, this part of the virus which is going to bind to the receptor, it will be covered by the antibody, IgA in particular, at the mucosal surfaces, and will neutralize the antibody so that it cannot enter, the virus cannot enter the cell. Then there are these CD4 T cells induce antiviral state and activate natural killer cell by producing IL-2 and interferon gamma, type 2 interferon. So we are now gone into the adaptive system, and now type 2 interferon are into play. They will perform their functions. Now back to our imagination. B cells, we need your help. So far T cells have been activated, CD4 has provided help to all the other cells. So what B cells can do, if B cells have been specifically activated, they have produced IgA antibody, or for that matter IgG antibodies. What they are going to do, they will actually play a better role during reinfection. Okay? Because during a viral infection, we are talking about the first exposure. This was the primary exposure. So during primary exposure, epithelial cells first, then comes the macrophages, epithelial cells, plasmacytoid dendritic cells, then comes the macrophages, they call the neutrophils, then further activation of the natural killer cells by the viruses, further adaptive immune system, CD4 cells, CD8 cells, they activate, the CD8, and then B cells are activated, they class switch. In the primary immune response, their role is not very good. But in the re-exposure, when the second time it comes, the memory B cells, they will produce lot of antibodies very quickly, and they will help in neutralization and complement activation. Okay, so in the reinfection, more stronger role is that of the B cells. So that's why you can see that memory will create, the memory B cells will be created during the primary immune response, and antibodies and CD8 cells against viruses. Now we will see that how they, both components of the adaptive immune system, how they are going to deal with the virus. So first thing is, this green color is going to show the protection, while red color is going to show the killing. So CD8 cell recognize the virus infected cells with the help of MHC class 1. It's going to produce the killing of this cell with perforins, granzymes, fast ligand, all those mechanisms. And they will also produce interferon gamma. And the interferon gamma is going to, create antiviral state and other functions. Antibodies, you can see these antibodies are neutralizing its receptor so that it doesn't bind to the cell and infect other cells. Natural killer cells, whenever MHC class 1 is absent or stress receptors are expressed, they will be recognized by the natural killer cell, and they also are capable of killing by perforins and granzymes. And they will also produce interferon gamma. Complement system can also be activated by the classical pathway, by IgM antibodies, and as a result they can sometimes also help in killing the virus infected cells, of course. And now macrophage role, you can see macrophage can recognize the virus infected cells or virus itself, take it up, phagocytose, and it needs help of CD4 cell, and it will give the help in the form of interferon gamma, and the macrophage will now be a better killer, and it will produce nitric oxide and TNF alpha. So this is all happening with the help of each other in a viral infection. Now these kinetics of innate and adaptive immune system are very good. So whatever we learned so far, you can see this is presented in this figure. So on the x-axis you can see from 0 to 12 are the days after the infection. Now immediately whenever the epithelial cells or any cells, nucleated cells have got the virus, the interferon type 1, alpha and beta, they start to produce and they reach the peak. And at the same time, natural killer cells, non-specific cells, they will be activated, they will start killing, so they will do their job. You can see the virus is starting to proliferate and it reaches its peak around 5th day. You can see here at the 5th day, the virus is reaching its peak. And at the same time, the adaptive immune system kicks in, and after 5 to 6 days, you can see cytotoxic lymphocytes, which are the specific lymphocytes, they are more increased in number and they are in the blood at increased levels. And finally, in the end, the antibodies, they come up the most end and then they stay longer. So these are the kinetics by which the one by one the innate and adaptive components appear during a viral infection. Now this person is feeling better because the immune system has rescued him from the infection. The student who caught the cold caused by this specific virus was exposed to the exact same virus 18 months later. What component of immune system will protect him from getting the same cold again? Memory B cells, because as I said, in the reinfection they have a very strong role to play, but that again, it is for containment, protecting its multiplication, protecting the spread of the viral infection. Again, elimination will be done by the cytotoxic T lymphocytes. Okay, so don't forget this point. They have a key role to play in containing the virus, stopping it from multiplying, stopping it from infecting or spreading, but the main elimination will be done by cytotoxic T lymphocytes. So the answer is memory B cells. And that's why comes in the role of the vaccines, that when you give vaccines to people, for example, against hepatitis B surface antigen, that is to produce in them antibodies, and whenever they are exposed to the viruses, they will try and neutralize the viruses. Now there is a simple concept that if immune system as a policeman, they find the virus or the thief, they are going to trying to eliminate or catch the thief and protect you, you are safe. But if the virus escapes or the pathogen or this thief escapes and hides somewhere, then you are not safe, there is a problem. And viruses have got certain characteristics by which they can cause a lot of problems and they escape your immune system. Now here in this graph of the WHO of 2017, thank you very much, you can see that the top 10 causes of death, here lower respiratory infection, you can see they have quite a big toll of death because of the lower respiratory infection. In particular, it was because of Middle East respiratory syndrome and dengue fever. Now why it occurs, this is just to signify the importance of viral infection, how important they are and how do viruses escape the immune response. There are three mechanisms. The strategies that the viruses use are avoiding recognition by host defenses. First is that immune system do not recognize the virus. Number two, play with the host immune system. Do something with the immune system that helps the viruses. Third is, even if they recognize, even if they are activated, resist the damage by the effector cells. So how it will happen, we'll see all of that. Avoiding recognition by the host defense. So what is the best thing? Viruses have got the property of altering its structure or antigen. They very quickly can alter their antigenicity, and best example here is influenza virus, then H1N1, they change. One vaccine will not be sufficient. The moment you make a vaccine, they transform, they change their structure. So in simple terms, they change their clothes. So how you will recognize them? Then latency. they do not replicate. Sometimes they hide somewhere for years. They stay dormant, and whenever they get a chance, your immune system has become weak in old age or because of stress or because of any situation, then they start to replicate and cause damage. Now, one of the example is, have you heard of herpes simplex virus? Herpes simplex infects in childhood, and sometimes it goes in the nerves and the ganglions, and it can stay for years, 20 years, 25 years, and in later stages, one dermatome is involved, it expresses itself as herpes shingles. That is one example. Another example is sometimes viruses will hide in immune privileged sites. Now, what is meant by that? There are certain sites, for example, brain, where excess of free lymphocytes, T lymphocytes, B lymphocytes, it is not, the luxury is not there. They cannot go like that freely into these sites. So some viruses will go and hide there, and the best example is that of rabies, which is spread by a bite of a rabid dog. That virus go and hide in the brain and then cause the infection. So you can see that how they play with your immune system. They do different things. Sometimes they will avoid the line of action. So wherever they think that we can be damaged, they play with that. Now we'll kind of start looking at some of the examples by applying where they can act. So you see their proteasome, which has the ability to break down the viral protein into small pieces that can be blocked by Epstein-Barr virus. So Epstein-Barr virus, it can block this proteasome. That is one example. Then another example we have talked too many times, MHC class 1. Its expression will be blocked here by HIV. So this is one example. This is another example. Then so downregulation of MHC class 1 is done by HIV virus. Then the proteasome is blocked, and it will not be broken down and will not enter the endoplasmic reticulum. There is a channel here in the wall of endoplasmic reticulum. It is called as TAP, transporter associated protein. Herpes virus, it can block this transporter. Okay, so you have got 3 examples here. Then you can see that interferon type 1, alpha and beta, which is produced by the infected cell to help the uninfected cell or the healthy cell. Sometimes what viruses do, they produce soluble receptors. So the interferon that is coming here, so there are certain soluble receptors that can catch this interferon type 1. For example, pox virus. Pox virus will take up, a kind of neutralize it like a sponge, take it, and it cannot go and perform its function here. Then when the interferon comes, it binds to the receptor and send signals inside healthy cells. Okay, these are the signaling mechanism. Some viruses can also block this signaling. paramoxyvirus. So you can see pox virus can block the soluble receptor, paramoxyvirus can block the signaling into the healthy cells, and then finally they can resist, so healthy cells, when get the receptors activated, they will kill the transcription of virus. But some viruses, for example herpes, again they can stop that as well. So you have got all these examples: herpes virus, Epstein-Barr virus, HIV, pox virus, paramoxyviruses. These all have got their own mechanisms of stopping the immune system from performing its function. Then most of the viruses, if you know that whenever bacteria is destroyed, viruses is destroyed, then the immune system has to be stopped, a check and balance. And one important cytokine in this whole process is IL-10. It is the regulatory cytokine, okay, remember that. And viruses know that. So what they do, viruses produce their own IL-10 to stop macrophages, dendritic cells, T cells, B cells, and they are called as VIL-10, viral IL-10. So you can see that they can produce, most of the viruses can produce viral IL-10, and that will inhibit the function of other immune cells. Then there is example in HIV virus, CD4 cells are destroyed. HIV virus directly enter into the CD4 cells and destroy a lot of CD4 cells. So again your immune system will become weak. So this is again about the inhibitory step, that pox viruses are there, paramoxyviruses inhibit the signaling and disruption of the messenger RNA step. And finally there are certain viruses that can also stop the effect of complement proteins. So these are the different mechanisms by which the viruses will escape the immune response. So that's all for this session. Do you have any questions? Yes, please. So how viruses affect CD4? So HIV virus, in your body you have got normally CD4 cells. These CD4 cells have got certain receptors, and HIV virus can directly enter through those receptors into CD4 cells. When go inside CD4 cells, every day lot of CD4 cells will be destroyed, millions every day. So when you don't have CD4 cells, they will not be able to help macrophages, B cells, natural killer cells, and CD8 cells. So automatically your immune response is very weak. Yes.
What was the function of IL-10?
Sorry?
What was the function of IL-10?